Good evening, everybody, and thank you so much for joining our webinar and our last webinar this evening. I'm Dr. Mercy Wanjala. I'll be your facilitator today. We are about to start, so we're just going to give a few minutes for everyone to join, and then we will start the webinar. We have about 40 plus participants online, and I think we can start off. So to open our webinar today, we have our KMA president, Dr. Simon Kigondu, here with us. Dr. Simon Kigondu, welcome to give the opening remarks. Over to you, Dr.
Yeah, thank you. Thank you very much to the team. And we want to welcome everyone to this final webinar of ours for the year, the collaboration between East African Medical Journal and the Research Committee of Kenya Medical Association. We hope that the lessons that we have been giving throughout the year have helped develop the research and writing skills of our participants. The next year, we welcome all of you to celebrate East African Medical Journal at 100 years, and this team will be in charge of ensuring that research continues in our country. We will be continuing with these research themes throughout the whole of next year, and we encourage all to join us at the Kenya Medical Association to promote scientific research. Thank you very much, Mercy, and over to the presenters.
Thank you very much, Mr. President. So over now to our program for the evening, where we are going to be discussing innovations in research. And to start us off today, we have quite a lineup of experienced researchers who are willing to share their expertise this evening. And to start us off, we have Dr. Maggie Sikulu Lord. Dr. Lord is an Amplify Fellow at the Faculty of Science, University of Queensland, Australia, where she currently leads the infrared surveillance and diagnostics team. Her research focuses on developing novel and rapid next-generation surveillance and diagnostic tools for detection of pathogens in humans, animals, and the environment, and for characterizing disease-carrying vectors. Her current specialization involves the development of non-invasive tools based on spectroscopy techniques. She pioneered the application of near-infrared spectroscopy technique for non-invasive detection of several pathogens in mosquito vectors such as Wolbachia, Zika, dengue, and chikungunya, and for differentiation of morphologically identical mosquito species. She has currently pioneered its application for non-invasive detection of malaria through the skin in partnership with the Australia Defence Force and Instituto Oswaldo Cruz in Brazil. Welcome so much, Dr. Lord. Over to you.
Thank you so much, Mercy. Thanks for that introduction. It was a long one. I was like, okay, that's really long. I know it's coming from Dr. Bonyo. Anyway, I will just share my presentation so we can start from there. Can we see my presentation? Can you see my presentation?
Yes, we can.
That's the last slide. I will go up to the first one. So I think I'll just start with a bit of introduction. I know Marcy has given us a very long and roundabout way of me, but in just a few words, I am a scientist at the University of Queensland, and I am also a lecturer. With research, I obviously specialize in developing novel tools for detecting infections in mosquitoes. And now I've just expanded to see whether the same tools can actually detect infections in humans. So today I'll be telling you a little bit about what I have innovated and how I see my research in the next 5 years or so. So when Dr. Bonyo asked me to talk about scientific innovations, I was a bit hesitant because I was just like, what do I actually talk about? So it took me a long time to decide what I should talk about. And it's just the other day that I thought, why can't I just talk about the work I do? So today I will talk about the work I do at EAQ. So how has research, how are we using research, how research is leveraging scientific innovations of all time. And we all know the internet, mobile phone, artificial intelligence, and medical imaging is one of the innovations of all time that, that have saved so many lives. And today I will mention how my research has leveraged some of these things to, detect infections in vectors. When I know I'm talking to medical doctors, I should have clarified that I'm not a medical doctor, I'm a PhD doctor. So some of these terms might not be familiar to medical doctors, but if you don't understand what I'm saying, just raise up your hand and I will clarify. So we are using a lot of internet and mobile phones and artificial intelligence, medical imaging to innovate in research. And how are we doing that with my work? So just a bit about mosquitoes. So how did this come about? So I actually did my BSc at the University of Nairobi and my master's as well. And then I did my project at Ifakara Health Institute in Tanzania. And that's where I met my supervisor from Australia. And I took up a project which involved telling the age of mosquitoes. And at that time, I was like, why is the age of mosquitoes very important? And it took me a very short time to realize that it's very important to know the age of mosquitoes. Why? Because when mosquitoes emerge from their breeding sites, it takes them about 2 days to seek for a blood meal. And once they have acquired the parasite malaria from someone who's infected, that parasite takes about 10 days to develop within the mosquito, which means a mosquito will not be infectious to someone until it's about 10 days old. Now, what we're doing is we want to find those mosquitoes that are, over 10 days old. Like you can see, those that are over here at the end with the red marks at the end are the ones that are carrying the parasites and they have lived for over 10 days. So that's what we are actually interested in. And the issue here we have is for us to know which ones are infected, we have to dissect the mosquitoes. And that takes us a long time. And there's only a few people who are interested in dissecting mosquitoes because you have to pull out their ovaries and check if they've ever laid eggs. If they have ever laid eggs, then you assume that they're potentially infectious. That takes a long time. So we want the interventions we have like bed nets to shift the age of mosquitoes to the younger ones so that the ones that are biting people are just nuisance mosquitoes. They're not carrying any infection. So that's why we need to know the age of mosquitoes so we can get rid of the ones that are carrying infections. And not just malaria, we do this for dengue as well. And because it's very hard to tell the age of mosquitoes, we decided to come up with a faster way of doing it. The near-infrared spectroscopy is like my baby. This is what I did in my PhD. So I tested the ability of using infrared to detect, to actually tell the age of mosquitoes first and see whether it can also detect infections in mosquitoes. That way, all we need to do is quickly scan the mosquitoes, know which ones are infected, where they were collected. And if we know where they were collected, then we know where the disease is potentially is, which means then we can go and spray that area, as opposed to just second guessing ourselves and saying, okay, people are infected, but where is the infection coming from? So that's the whole point of doing surveillance, collecting mosquitoes. So the near infrared technique is very attractive for a few reasons. Firstly, it involves just shining a light on a sample. So it's figure out how you use a torch, you know, how you point a torch in darkness. That's how infrared works. So you point a light onto something like a mosquito, you get infrared signature, you use machine learning or artificial intelligence, like I mentioned before. And then you can tell the difference between, you know, those that are young, those that are old, or those that have infection. So I pioneered the application of infrared for detection of infection in mosquitoes like Zika. Firstly, the age grading, first we can tell which ones are younger and older. So this is just an spectral signature after we scan mosquitoes. That's me scanning mosquitoes. So we're just pointing a light, we collect this spectrum, and this spectrum tells us which mosquitoes are infected or how old they are, if they're infected with Zika. I pioneered its use for Zika detection when there was an outbreak of Zika in Brazil for the first time. And the other thing, we have also shown that we can detect American trypanosomiasis infected, not infected mosquitoes, but infected triatomine species. So we've shown, so many applications of this technique, and the next step is now to, you know, take it to the field and see how, you know, this can be applied to detect hotspots. Okay, let's move to the next slide. So what happens next is, yes, we can detect infections in mosquitoes. And I was so curious, can we use this technique for something else? What you're seeing here is actually mice infected with malaria. So I wanted to know, I was very curious, if I can detect the infections in mosquitoes, can I actually detect malaria as well? Because as we know, malaria is in red blood cells. But obviously I can't just go straight into the field to scan people. So we started off with infecting mice in the lab with malaria and, you know, shining a beam of light on them to see whether we can detect malaria. And as you can see, we just pretty much a torch, shining on a mouse tail or the foot or the ear. We collect the same spectral signature. You can see here, those that are not infected are in blue. And those that are infected are in these different colors. And we can tell those that have high paracetamia and those that have low paracetamia. And this was very exciting. So then the next thing we did was to just see how does paracetamia affect prediction. And we saw that as the paracetamia of the mice went up, the sensitivity of the technique also went up. As you can see here, paracetamia is over 3%. We get over 95% accuracy. And this is when we are scanning blood as well as through the skin. So we wanted to compare if we just scan blood directly and if we scanned through the skin, do we get the same results? And it's pretty much the same result. So that was exciting for us to see. And this work here then prompted us to see, can we take it to humans? And actually, I'm very happy to present these results to you. This paper has just been published 4 hours ago. So it's very exciting for me to share with you just as it has been published. So we took the work to the field and scanned human beings. We scanned their fingers, those that were infected with malaria and those that were not infected with malaria, to see whether our infrared technique can detect the infections. And the result was very good. As you can see, here we are scanning a finger, and here we're just scanning an arm. We couldn't show photos of the ears because of the ethics, but the best detection came from the ear. I don't know why. I don't know if it's because the ear is cleaner and the light penetrates further into that. But yeah, you can see that the spectra is different for those that are infected with malaria and those that are not infected, or those that are infected with different species of malaria. And these are just the peaks that are connected to malaria infection. But we also wanted to know whether we're actually just detecting, noise, or if it's something related to Plasmodium. So we scanned purified Plasmodium parasites and compared the peaks. And so you can see these peaks actually match the ones that we acquired non-invasively when we scanned purified parasites. And that's very exciting. And so the next step here is to take the work to the field. So you can see the sensitivity for scanning the ear was 100%, and specificity was 85, but this is when we have run the artificial intelligence, and then we use the model that we have developed to predict the ones that were not put in the model. So this is an independent data set. And those two people that were not correctly predicted were the same for year 1 and year 2, which means that we collected the spectra with minimal error. So that's very exciting. And now we want to work with other countries to expand the study. And I'm actually here to do the field study. We're doing the work with KEMRI in Kisumu. We want to see if we scan people, can we actually detect malaria and other infections? Something that I haven't mentioned here is from funding from the Gates Foundation. We've also shown that we can detect tracheories, the parasites, through the skin of mice. And it'd be interesting to see if someone is infected with both malaria and tracheories, if we can detect the two parasites, or if we can show that we can differentiate people that are co-infected with soil transmitted helminths and malaria. And then the next steps, obviously, is to validate this in multiple field settings where there's mixed infection, skin color, testing in children and pregnant women. And we also want to optimize the scanning protocol because we're not really sure whether it's just the ears that gave us sensitive results or highly accurate results, or whether we can actually scan the foot as well. We want to expand the areas that we scan and see if we can improve on that. And I just want to touch briefly on a tool that we have just launched as well. So obviously, if this works, we would like to integrate it into testing into a disease advisor platform, which I'm going to quickly mention in my next slide, and explore the possibility of wearables. And this is where can we just use my mobile phone to scan my finger, or can I wear a ring that has infrared light, and whenever I get sick, it can just, ping me and tell me, hey, you need to go see a doctor, or even in watches as well. That is the next innovation. We are not there yet, but I have a dream that we can actually get there. So disease advisor platform. This is something that we just launched. It's actually outside my University of Queensland work, but I thought you being medical doctors, you might be interested in hearing what this platform does. So just from the name, Disease Advisor wants people in real time when they encounter infectious diseases. So for example, if you're traveling from Nairobi, you're going to Kisumu, it tells you you have just arrived in a hotspot of malaria or other infectious diseases. It also tells you the causes of the diseases, how to protect yourself, and whether there's doctors around you and things like that. It can send you alerts about outbreaks in the area you are in. And we have also incorporated a feature where we can work with businesses like medical doctors and, travel doctors and pharmacies and hospitals, so people can reach out in the area within that disease hotspot. Obviously, the most important thing here for us to remember is disease advisor tells us what is in the area we are in, like right now at this particular spot, what sort of diseases are here. Let's check it out. I'll just click on it. Can we see this other screen?
No, we are still seeing the slide.
Are you still seeing this slide? Okay.
Yes, you probably can share the other screen as well.
Okay. But is it opening? Let me open it first. Yeah, there you go. So I probably will need to stop sharing this one and then share the other screen. So this is Disease Advisor. It's a web app for now. The mobile app is being launched in the next 2 or 3 days. You just need to fix a few things. So what it does, assuming we are going to Kisumu, or you just need to type here is Kisumu. Can you see this screen, Marcy?
Yes, we can now see the screen. Thank you.
Okay, so for example, if I'm going to Kisumu, it pops up with those diseases. It says Kisumu is at risk of dengue, malaria, schistosomiasis, and Anopheles mosquitoes and other soil-transmitted diseases. If I click on it, it tells me the causes, symptoms, treatment, vaccination, protection. And the other cool feature here we have here is, you can see we have like travel doctors, and right now it is going straight to Google. But what we want to do is we want to work with doctors who actually want their work featured here. So we can only list those doctors that just want to go back to the other screen. That want their businesses listed here instead of us just listing everything. We also have, we're going back to the screen. Yep, so we'll go back here. So we also have pharmacies here. If I click on pharmacies, it takes me to pharmacies. Hospitals, takes me hospitals. Hotels, we want to list only hotels that are safe for people to stay, or restaurants that provide, repellents and things like that. Public transport that have, are safe for people, because it's all about protecting people. And the other thing that you as doctors might like is we have, I don't want to sign in now because it's going to take more time, but you can play around with Disease Advisor at your own time. When someone has traveled and they decide, okay, I don't want to use the app, I'll just leave it on. When they go back to where they came from and they start feeling sick, they can go back to their travel history, and then we can show you that this is the disease risk history for you, because you are in Kisumu, you probably were exposed to these diseases. And when they come to you, it's easier for you to actually diagnose them based on the symptoms and based on where they came from. And I think that's the end of my presentation. I will answer questions later.
Thank you so much, Dr. Lord, for that wonderful presentation. I think this is quite groundbreaking innovation in research, because these are the realms that we foresee ourselves going, and especially in an area in tropical diseases. So thank you so much for that wonderful presentations. We'll take questions for Dr. Lord after the presentations. So we are now going to move on to our next speaker, who is going to be taking us through innovations as well. And we have Dr. Obonyo, who is no stranger to us in KMA. He co-chairs the KMA Research Committee, and he holds a doctorate in cardiology and is currently a postdoctoral cardiac research fellow at the Critical Care Research Group based at the Faculty of Medicine, University of Queensland, Australia, where he has also been involved in research response to COVID-19 pandemic. He is a visiting academic fellow at the Institute of Health, involved in biomedical innovation, medical engineering research facility in Queensland, University of Technology, Australia, and he is also an alumnus of the initiative to develop African research leaders based at the Kemri Wellcome Trust Research Program in Kilifi. Welcome, Dr. Obonyo, over to you.
Thanks, Marcy. I hope you can hear me and you can see my screen.
Perhaps, I don't know, is there a way you can increase your volume just a little bit?
Let's see, is that better? Can you hear me?
Yes, we can hear you.
Okay, perfect. Thanks for that introduction. The stuff I'll present is much less cool than what Maggie does. And I took the liberty to just walk us through clinical innovations in general. Then at the end, I'll just talk briefly about the work that I do. Going back to what is the definition of clinical innovations, in this paper in 2017 by Drs. Kelly and Young, they say that innovation is a newer, better, more effective way of solving problems and can combine either an invention, an adoption, or diffusion of a technology that's already in existence. When you think about clinical or healthcare innovations, it will be a novel idea, product, service, or care pathway that has clear benefits when compared to what is called standard of care or what's currently being done. The major characteristics of clinical innovations is that they need to be usable and desirable, of course, paying attention to the principal doctrine of medicine, do no harm first. If we look at medical, historical perspective of medicine from the days of Hippocrates, the clinical innovations have been happening throughout the spectrum of medicine, and you can end up having a webinar that goes on endlessly if we are to talk about clinical innovations in general. So I'll briefly focus on, so these are 7 medical breakthroughs that are built to have changed.
Dr. Obonyo?
Yes.
Could you please, yeah, you need to increase your volume. You're not
okay.
loud enough. Yes.
Okay, sorry. My volume is 100%. Okay, I hope that's clearer now.
That's better.
Yeah, so these 7 medical breakthroughs built to have changed the way medicine is practiced today, starting with Ted Jenner's invention of vaccination. We are just coming out of a pandemic where COVID had stopped the world, and it's just when we got the vaccine that we were able to resume some level of new normalcy. Practice of anesthesia, invented by William Morton, epidemiology by John Snow, germ theory and asepsis by Joe Lister, insulin by Sir Fred Banting and colleagues, and then 3D printing started by Chuck Hull, and biomaterial printing later, and then gene therapy by French Anderson at the beginning of the 1990s. When we work in our practices day to day, there's some innovations that happen that we can almost take for granted today. So there's this article by this guy called Kashyap Vyas that talked about 13 medical inventions that have changed the world. So I'll quickly just remind us of each of them and highlight towards the end how we are improving on some of these or working towards improving some of these in the lab here. The first one, of course, mercury thermometer, started with that simple one and moved to digital ones. Then René Laennec, a French physician, introduced the stethoscope, which just amplifies sounds. Then Konrad Röntgen introduced X-ray imaging. Introduction of spectacles in around 1800s, when there was increasing literature or reading material. It's not really attributed to any particular person. However, the University of Pennsylvania, this was earlier this year, just published this a single injection using an experimental RNA technology into the eye can reverse blindness in patients who have genetic disorder. This is just, I found this very interesting when thinking about spectacles. The other invention that has changed medicine is, of course, antibiotics, started by Alfred Bartham and Paul Enrich, that started as Salvarsan. I think today is in the market as Phenamine, and then in the 1920s, Fleming introduced penicillin. The other interesting thing about these drugs, of course, we did mention about 3D printing. Now, I mean, FDA, this was in 2015, actually, FDA approved the 3D printing of pills, and who knows what the future holds. Hypodermic needle, we use this every day in our practice, introduced by Charles Pavas and Alexander Wood. Disposable catheters have completely revolutionized how we do dialysis and cardiac catheterization. The cardiac pacemaker, introduced by Mark Hill and Edgar Booth, these are actually Australians, and they use this to resuscitate a young boy. Of course, it was slightly bigger, the size of a fist, and has undergone lots of technological advances and innovations, and now looks as what you can see at the bottom there, that was implanted as a wireless device in a patient in Bismarck. This new wireless pacemaker actually reduces complications and has battery life of almost 20 years. So it's deployed into the heart. There's no wires, no cables connecting. It just releases signals to the heart. Again, CT and MRI, CT by Dr. Geoff Honsfield, MRI by Raymond Damasian, and then the new molecular breast imaging for detection of early breast cancer. Number 11, Claude Beck introduced the cardiac defibrillator, which he used to resuscitate an 11-year-old boy. Nowadays, automated cardiac defibrillators are almost everywhere, for use by the public. The other invention is a total artificial heart. This actually is technology that started way back in 1912 by Le Galois, but then it's only 170 years later when Dr. Jarvik revolutionized it into what it is nowadays. As you can see, there's a left and right-sided pump with tubes connected to a bag which has a battery pack. But then this is actually an older model, and this is part of what the lab that I'm currently based is trying to work on improving. The last one is bioprosthetics that have revolutionized medicine, where people have, as a result of either diabetes or mangled extremities, undergo implantation of prosthetics prostheses that tend to perform as good as original limbs. I'm going to change gears a bit and just bring this into perspective. This is of interest from my own clinical work. We had a pig heart transplanted into humans successfully for the very first time. This was done after a lot of research that involved gene editing from the donor pig and inactivating pig genomes, and this was conducted by Dr. Bart Griffith at the University of Maryland. The patient lived for 57 days, but this, you can imagine what this does to the field of heart failure, where the definitive treatment has been organ transplant. Is of course not enough. donor hearts going around, and if we can use just genetic tissue engineering to develop grafts, then it just changes how we'll be treating patients in the future. Gene editing is not new. The first, cloning actually started in 1996. For those who remember, this is Dolly the sheep that was cloned in Uni of Edinburgh at the Roslin Institute, and that's just a scheme of how that happened. Now I'm going to just finish up by showing slides of the lab here and what we do. So talked about the total artificial heart. This is a newer version which uses a magnetic levitation technology. You have the right side and the left side, and inside is just a spinning disc, spinning magnetic disc that, as volume changes from the right to the left side, it can also shift upwards and downwards. That's what is being displayed there. So you have the right side, the top with the blue blood, and the left side down there with the oxygenated blood. And this uses just a magnetic levitation, maglev technology. As you can see there, if there is more volume on the left side, the magnet shifts to the top, or if there is more volume on the right side, the magnet shifts to the bottom to accommodate more volume. So this is revolutionary, and this is just how it gets implanted and how it sits in the heart. This has already been tested clinically. I don't know why we use sheep, maybe from the days of Dolly the sheep. This is a sheep in the lab that has been implanted one of these devices. It is pulseless, but you can see, it's having its normal feed. You can tell it's post-op with its tubes there still hanging from the neck, but otherwise very normal. This lived for about 7 days, and it's courtesy of the Critical Care Research Group Bionic Heart project that is currently running in the lab here. Other innovations going on in the lab. This is my boss, Professor John Fraser. So the big project we're doing is the ICU of the future. This is actually going to be launched next week, so I'm not really allowed to show the actual picture of it. I just have a scheme of what it will look like, but it's quite clever if you think about it. When patients are in ICU, they tend to have all these loud alarms at the back of their head, which is not really meant for the patient. It's meant for the nursing or medical staff looking after the patient. So the ICU of the future has used noise reduction technology to make the patient environment as comfortable as possible. And I think it's something good for the future. The other thing that is happening, when the pandemic started, COVID-19, there was, no one knew how to treat it. So the research group here started what is called the COVID-19 Critical Care Consortium, something that just started as a call amongst colleagues in working in critical care, intensive care across the world, ended up being the largest network of intensive care units. So there's currently over 400 ICUs in the world within this consortium in one platform. And this has also speeded up discovery of new treatments or treatments for COVID patients. The other thing, can remember during COVID, patients, or when clinical staff are wearing hazmat suits, the risk of transmitting infection. One of the new innovations that came out of the Asia group was, this is actually quite interesting. They used technologies that adopted voice technology. I think the Alexa, Siri, instead of going with a file around the patients while doing a ward round, that time you're dressed in a hazmat suit, you just shout to a device, the temperature in this patient is, whatever, 39 point something. And that voice signal gets adapted into a digital signal. And they've now taken it even a step further, where all this data now for research purposes, as you can see on the slide there, gets easily translated or coded from the signals you have on the monitor into data that can be used in a research environment. You can see that has just been digitized into a spreadsheet or a database that you can make use of. The last thing I want to show, so this is a dashboard where, as I talked about the 400 platforms, as you can see at the top there, the dates counting upwards to, and the sites across the world, the different ICU sites, just showing the number of patients with COVID as the database gets populated. This is the first of its kind, again, just part of an innovation that comes from the Critical Care Research Group alongside all those partners listed at the bottom there. The last thing I want to just mention briefly is this is not too far away from us. It's right at our doorstep. The KEMRI Wellcome Trust program, together with the Initiative to Develop African Research Leaders, collaborates internationally with this group, and that's actually how I ended up getting here. And so the opportunities are there right at our doorstep. I think that's the end of my presentation. I just wanted to thank all of KMA, National Executive. When we started these webinars in the middle of COVID in 2021, we didn't know what this would look like. We ended up we've had one webinar each month, starting from 2021 there all the way, and this would be the last one for us this year. And I want to sincerely thank the membership, all the presenters and speakers who have very generously donated or volunteered their time and shared their knowledge and made this the success that it is. I want to end by just leaving us with the Royal Society motto that clinical research is not easy, but it needs some level of inquisitiveness to do. The motto there says nullis in verbra, which just means take nobody's word for it, do an experiment and come up with your own results. Thank you very much.
Thank you so much, Dr. Obonyo, for that scintillating presentation. I mean, the evening just keeps getting better and better. At least now, when someone says medlev technology, I'll know what, maglev technology, I'll know what that means. And I'm really looking forward to seeing more on the ICU of the future, because I believe a lot of us have always had a problem with the noise that the machines produce, and it's very damaging to the patients and deters their healing as well. So we look forward to looking at that. And to wind up for us today, we have another speaker who is Professor Ambrose Aguayo, a pediatrician and clinical epidemiologist with broad interest in pragmatic clinical trials, childhood respiratory illnesses, and clinical guideline implementation. He's a professor at the London School of Hygiene and Tropical Medicine, a principal investigator at the KEMRI Wellcome Trust Research Program, where he, until recently, was the head of Department of Epidemiology and Demography, and also holds an affiliate professorship at Stanford University in the USA. Nationally, he serves on various Ministry of Health technical panels, where he has been involved in the development of the national policy and strategy documents for newborn, child, and adolescent health. He is also the co-chair of the WHO UNICEF Child Health Accountability Tracking, that's CHAT, Technical Advisory Group. Thank you for being with us this evening, Professor Ambrose. Over to you.
Thank you, Mercy, and good evening, everyone. I think my role here is to moderate the discussion from the talks which have been so ably presented by Drs. Lord and Obonyo. It's really inspiring to see the amazing work that homegrown talent, those who were trained in environments similar to our own, are doing out there and really flying our flag high. So thank you very much for those inspiring talks. I want to check if there are any comments in the chat first before I pose some questions for the speakers of my own. And maybe just to start with, there is one question from John Masasabi asking about the performance of the diagnostic malaria diagnostic tests that use near infrared technology in dark-skinned Africans. Lord, do you want to quickly?
Yes, I'd like to, yes, that's one of the key things that we actually want to assess in our scale-up study. Skin color is obviously going to play a big role, which is why we're aiming for parts that are not, not interfered with skin color, like inside finger or inside toe. We did scan patients in Brazil, in the Amazon, and there was a range of colors there too. There was the dark skinned and there was the lighter skinned one. And what we did, the beauty of artificial intelligence or machine learning is you can actually regress out the skin color. But the sample size was very small, so we don't want to make a call on the skin color until we have scaled up the study. What we might end up doing is we do a study in an African country, do a study in Asia, and try and see how this compares. If we can put them together and regress out the skin color, that will be good. Otherwise, what we could do is just have separate predictive models for the Asian people and the African people. But it'd be interesting to see how we can do this. But we did manage to regress out the skin color in the current study.
Wonderful. And I have a linked question specific to that aspect around the performance of the diagnostic tool which you've innovated. And I was really impressed by the high sensitivity of 100%. I know that the publication has just been released, and congratulations on that. Could you please, though, speak to the characteristics of the reference population among whom this near-infrared technology was compared to generate your sensitivity and specificity estimates? What was the gold standard?
So we did, obviously, firstly, I want to say something about the general objective of this tool. I really actually do not want to use it as a diagnostic tool per se. I want to use it as a surveillance tool. Reason being, we want to identify the asymptomatic patients because they're the ones that mosquitoes like so much. Mosquitoes will bite them and they pick up the infection from them and then go bite the small kids, and small kids are the ones who get malaria. But the problem is, if they're not feeling sick, they're not going to come forward to get tested. So that's the issue we have. So if we can actually convince them to, not give a blood sample, but just quickly get scanned through the skin, we probably will be able to try and track them down and get them treated so that we can reduce the level of malaria. So I'm interested in using it as a surveillance tool so we can scan thousands of people, say in a village. I come from Sirisia, where Dr. Masasavi also happens to come from, so he's my village mate. So the population that we tested the tool is Brazil, in the Amazon, and we had people with high paracetamia. Like a few people had very high paracetamia, but most people had very low paracetamia. And what we only wanted to know is, can we differentiate those that are infected from those that are not infected? Because if we go out in the field, we want to catch everyone that has malaria. So we didn't care really the paracetamia level. We just wanted to know, are you infected with malaria or not? So firstly, we did microscopy, but we knew because there was a lot of low paracetamia, we might miss out some. So we did PCR. We didn't do quantitative PCR though, because we just wanted to know who is infected, who is not infected. It took so long for us to publish that paper simply because we didn't do quantitative PCR, but we did manage to convince this PNS Nexus journal to publish the work after a long time.
Wonderful. And I'm sure the clinical folks in the room are wondering at what point will they have these tools at the bedside. But what you've suggested is that the utility will be more for surveillance than for bedside use as a diagnostic.
Of course, I would like it to be a diagnostic tool as well, but I obviously don't. If you miss out 2 people, 3 people, that's really bad as well. But if you miss out a few asymptomatic patients, it's not as bad. So to start with, I would pitch it as a surveillance tool, so we don't feel too bad if we missed out a few asymptomatic patients.
Wonderful. Thank you. Thank you for those responses, and really waiting eagerly to see these tools come to mainstream use. Obonyo, thank you for your talk and a very fascinating overview of what medical innovations are, and then zeroing in onto some of your personal interactions with innovations in healthcare. My question is slightly philosophical and bordering on both philosophy and medical ethics. These innovations, especially those which are more recent, gene editing, for example, are often presented as tools to solve medical problems, medical challenges. But one can envision their utility extending to enhancing performance of healthy people. And that obviously comes with its own caveats as well. I'm sure some have heard about this Neuralink technology, which Elon Musk is very ready to roll out, inserting microchips in individuals' brains, which would have applications for individuals with neurological disease. But one can imagine that it might be used to enhance cognitive functions among healthy people. Any comments around this, and whether we're ready for some of these innovations that are very close on the horizon?
Yeah, thanks, Ambrose. I like when you said it's a philosophical question, because I don't really have a straight answer to it. But of course, there's lots of debate around the ethics of lots of these innovations that are coming up. I think it was the US President George Bush who even banned funding from the US government for any stem cell work. And this was around about just after Dolly the sheep had been cloned, and there was a lot of talk around it. And he said, no, this is now playing God, to just borrow his words. So that there's usually lots of, I think, ethics considerations and discussions. And I think that just goes back to the Hippocratic oath that we normally take in terms of who stands to, who should we offer these therapies to. As you rightly point out, performance enhancing can be a very big problem in the middle of the World Cup, and you can imagine if someone gets performance enhancing and then they end up lifting the World Cup or winning a marathon, that just, you know, changes how sports has been. And so I think while the technology is coming out, I think as medical professionals, as healthcare researchers, as policymakers, everyone, all stakeholders need to be involved in developing both legal frameworks and, you know, how this should be rolled out and how it should be applied. Probably the last, what I should mention probably is, I think what I personally strongly feel is, I think that there's also the bit that is unethical in not offering patients an opportunity to participate in research. I think offering patients, it should be upon the patients to say, yes, I want to take part in research or not. Not offering them that opportunity, I think also borders on unethical. Thanks.
Yeah, thank you, Obonyo. Very balanced response there. And I know we could go on discussing this for hours. I can see a few comments in the chat, I think directed at Dr. Lord. What's the effect of different hemoglobin levels on the infrared diagnostic tool? And maybe if you could take this along with the second one, what are the confounding factors to the infrared light technology in malaria detection? is the main aim for this technology for screening purpose or diagnostic purpose? I know you've already tackled a few of these. Maybe just hand over to you to quickly summarize.
Yeah, so the first question was, what was the first question, sorry?
Anthony Wanyoro is asking, what's the effect of different
hemoglobin. Very good question. Yes, it has an effect. And when I actually wanted to test this, it was mainly because I knew the hemoglobin concentration is reduced in malaria patients. So my first biomarker was, I need to look for hemoglobin in patients that have malaria, and that will be the first biomarker that these patients are infected with malaria. If you saw the spectral signature that I showed you in my paper, I have explained that the reason why we could differentiate infected from uninfected people was, number one, the hemoglobin concentration was different for those infected and those not infected. That was one of the reasons. And the other reason was we could detect the proteins of the parasite. So hemoglobin concentration is number one marker. And that was what I was looking for when I actually innovated this technique for malaria detection.
Thank you. And any other confounding factors to the
skin color? Yeah. Yep. So the skin color will be one of the issues. I think age is also going to be an issue. And the other thing could be if you're infected with other infections that affect, the blood as well. Can we actually accurately tell that this person is infected with malaria and another infection, or is it just going to, put it all together and say, yes, you're sick, go and see a doctor? And this is why we want to scale up the work. We are in discussion with the Bill and Melinda Gates Foundation. They're interested in looking at all these different factors that could affect the sensitivity, and is it, specific for malaria, or is it picking up other things as well? So yes, the question is, this is early days. We don't know yet. We know there will be these things will contribute to sensitivity, and we want to track them down and say, yes, this peak here is for malaria, this peak here is not for malaria.
Wonderful. Thank you. And I think, we're at the top of the hour. We could go on and on with questions. I can see a few others coming in the chat, but Obonyo?
It's a very interesting point Dr. Antihava has just raised that I could probably just quickly respond to, where she's asked about ownership of patency for collaborative innovations. I think there has been a lot of debate around this, and in 2009, I think there was the Working Party, and this was organized by the Organization for Economic Development and Partnerships. I think there was a meeting in Paris, and that's usually the guideline or the guiding document that is used for this. So very similar to the way we use the EAMJ guidelines for attributing authorship in publications. There is very structured mechanisms for deciding patents, or who owns what proportion of an innovation. In terms of financial resources localized to global space, I think part of this is probably a discussion that transcends my expertise, but I think it's about time we stop thinking of just asking for vaccines. If you've just seen the platform that I shared when COVID was evolving, we had almost 100,000 patients' data. The vaccines are not made by aliens from Mars. They use this clinical data to actually inform the development of vaccines. So we can actually contribute to the development of some of this with minimal financial resources, but using the population information that we have. I'll probably stop there. Thanks. Thanks very much, Ambrose, and congrats, Maggie, on that publication.
Thank you so much for having me. That was a pleasure presenting to medical doctors. And you should come to Australia.
Thanks. And obviously, if you're in town for a while, there might be those who might want to reach out to you directly and speak to you in person. Very proud of both of you, and thank you very much for the talks.
And I've just put my email address into the chat for anyone who wants to reach out for questions. I'm happy to get in touch with them directly.
Thank you. And also in the chat, I can see a link to a AMA Mental Health Needs Assessment Survey. Please take less than 5 minutes of your time to feed that back. The form it's a Google form with a link provided in the chat. I will now take this opportunity to invite Professor Okeo, who is the board chair for the East Africa Medical Journal, to give us a vote of thanks and close the meeting. Thank you very much.
Thank you, Ambrose. Good evening, everyone. I want to appreciate very much Dr. Sikulu's innovation presentation, as well as Dr. Obonyo. I actually had a question of my own. If you agree, I will post it, and then after a response, I can go on. And I wanted to say that I think a couple of days ago, actually last week, coming back from Cape Town, I found myself with an app on my phone, and it was requesting me to do stuff. It had to do something with COVID. And I suspect this is also an innovation that I thought came up in the middle of COVID-19, where you could identify hotspots for COVID. So I just wanted Dr. Sikulu to draw a parallel between, because it comes out of an innovation on surveillance of COVID, to find out where COVID is and where it is not, so that one can take appropriate action if they are able to. Dr. Sikulu, are you able to do a parallel?
Yes, I can. Professor Okeo, thank you so much. So Disease Advisor was actually born just before COVID. So it was very hard for us to convince people that, hey, this is not born out of COVID. So the idea was conceived, and we actually put in our IP just before COVID hit. And I'm sure if we waited, a bit longer, we would not have put in our IP, because it would never be innovative. So we don't do COVID because we thought, well, COVID is everywhere. Disease Advisor is only, talking about these other diseases. We can tell you whether there's an outbreak of COVID in an area or there's excessive outbreak of COVID, but we're mainly focusing on these other infectious diseases. We tell there's an outbreak of Ebola, cholera, but the main thing is we tell you in real time if you are at risk of contracting any other, infectious diseases. And we're not doing contact tracing like other COVID apps that would, tell you that you came in contact with someone. No, we just tell you whether you are at risk and what you can do to protect yourself and what sort of help is around you and how you can reach them. Did that answer your question? Professor Okeo?
Looks like his network.
Dr. Obonyo, did you hear me?
Yeah, I did. I think Professor's network is off.
Or dropped out, okay.
Maybe Ambrose, you can just...
Yes, of course. Unfortunately, we've lost Prof. Okeo, but I think we were able to hear his question and Dr. Sikulu's response. This is just to thank, first of all, the organizers of this webinar, Dr. Obonyo was thanking a team of individuals at the end of his talk. I think he forgot to thank himself. Obonyo has really championed the work that the Kenya Medical Association Research Committee has been running alongside the East Africa Medical Journal with these webinars. And I think one big hand to him and the team on the committee which he leads. I also want to recognize the role of the EAMJ and East Africa Medical Journal leadership for providing that conducive environment for these meetings to take place. As we close the year, it's been quite a successful venture, and we look forward to many more in the new year. So thank you for your leadership, specifically Dr. Kigondu, for being there and showing your stewardship and support for these activities. Speakers, of course, thank you for making time for this webinar, taking time to prepare presentations that were highly stimulating. And once again, just expressing our pride for the work which you're doing. I'm sure you remind people that you are Kenyans out there doing your wonderful work. So we're very proud of you, and thank you for presenting to us. Then, of course, to the audience, we would be speaking to ourselves if you were not there. Thank you for making time in your evenings and for your stimulating questions, and even those who didn't ask questions, I'm sure you've left here with some additional knowledge. I'm reminded that this is a CPD accredited activity, and those who attend will be able to claim your continuous professional development points from this. I can see Professor Okeo has managed to rejoin. Prof. Okeo, I took the liberty of giving a vote of thanks, but I'm sure the audience would love to hear from you.
Just go ahead and complete. Sorry, technology let me down there. Please go ahead and wrap it up. Thank you.
All right, thank you. And I think I'm done. I just want to wish you all a wonderful evening. And I believe this is the last of the series for the year. Obonyo, is that right?
Yes, that's right. Probably if there's any suggestions that for improving this, they can be channeled through the KMA Secretariat, Dr. Mercy Wanjala as well, and we'll be more than happy to take up any suggestions for the coming year. Otherwise, from me, I think it's a Merry Christmas to all of you. And thank you very much.
Thank you, Obonyo. And Dr. Wanjala, I don't know if there are any administrative final remarks that you might have. Dr. Wanjala, you're muted. I'll assume you're...
oh, sorry. Yes.
Any final remarks from you, administrative or otherwise?
No, I think for me, it's just to thank everyone, the team that has really done a wonderful job the whole year. And also for this one, you as the moderator, all the speakers who are giving us the expertise for free, which is an amazing thing, just sharing and also trying to grow young researchers in Africa. I am also a beneficiary of this, even as a member of the research committee. And that is all. And hopefully we will see you again next year as we start another series.
Wonderful. And of course, thanking you, especially Dr. Wanjala, for making this happen. Have a good evening and happy holidays to you all. Be blessed.
Thank you.
Thank you. Happy holidays.
Bye.
Bye-bye.